within our animal diversity, the first phylum that we have to know about is the phylum Periphera. The common name for Porifera is our sponges, and these are aquatic marine species. What's important to note about phylum Periphera is that they are the basal taxon, which means that they were the first phylum to diverge, and they are most closely related to our ancestral protist. So the phylum Periphera diverged early in our evolutionary history of species. Now, there's two characteristics that are unique to Periphera, and that is they have no true tissue and they are asymmetrical. These two characteristics are only exhibited by the Periphera in all of the other species we are going to learn about. Since the phylum Periphera have no true tissue, they are referred to as Perizones. Perizoa simply means no true tissue. The opposite of Perizoa is Eumetazoa, and Eumetazoa means true tissue organisms. So the Periphera, they have no true tissues, therefore they are Perizoans. They have no symmetry, they are asymmetrical, and they can either be a few millimeters large or up to a few meters large. They have no coelom. For locomotion, this is very important because the adult sponges are sessile, but the larvae are able to move. So what happens is that when we get this larvae sponge, it is able to swim to find a suitable substrate. Once it finds a suitable substrate, it will attach itself and then it will continue to grow into its adult stage. And once it reaches that adult stage, it will be sessile and no longer motile. So since we know that the larvae is able to swim, that means it must be flagellated, because remember, a flagellum is used to propagate that motion, especially in an aqueous water environment. So locomotion, important to know the distinction. Adults are sessile, larvae are able to swim. For feeding strategies, the phylum Periphera are suspension feeders, which means that they filter out food particles from the surrounding water. And we'll see exactly how that happens. But they are suspension feeders, and we have two different types of cells that are very important when we talk about feeding, and those are the choanocytes and the amoebocytes, which we will also get into detail about. In regards to reproduction, they are sequentially hermaphroditic, which means that they change their sex at some point in their life. Since they are hermaphroditic, that means that they are able to produce both eggs and sperm. And since they are able to produce both egg and sperm, that means they are able to sexually reproduce, or they can asexually reproduce. And remember that sexual reproduction can either be self-fertilization or cross-fertilization. For the asexual reproduction, we can form another sponge through budding. And budding is essentially when you have that parent organism and you see a cluster of cells begin to propagate from that parent organism. Once that propagation of cells continues to grow and grow and grow, it will detach itself and we will have a new Periphera. And that's budding. So since there is no true tissue, we have to understand that there can't be a nervous system or a respiratory system or an excretory system. Why is that? Well, what do we know about tissues? We know that tissues are a group of cells with a similar function coming together to create some sort of a system. Now, if we don't have tissue, we can't have the organs to carry out these processes. So since there are no tissues, we won't have a nervous system. So that means if a fish or another organism is interacting with the Periphera or the sponge, it will not be able to detect whatever is interacting with it because it does not have a nervous system. For respiration and excretion, that's done simply through diffusion because all of the cells on the Periphera or on the sponge are in direct contact with the water. So since all of the cells are in direct contact, they can simply respire and excrete through diffusion. While we're on the topic of the cells, it's important to also note that the Porifera have two distinct cell layers, the outside layer and the inner layer. And the center is a gelatinous matrix, which we refer to as the Mesohill. So Mesohill. It's important not to confuse this with mesophyll. Those are two different things. Here we have a mesohill, which is the gelatinous center that separates the two cell layers. And the mesohill will be important because that's where we will see the movement of the amoebocytes occur. So everything on this page is very important to understand about Porifera. What's really important to know is that it is the basal taxon. It is the first to diverge. It is asymmetrical. It's asymmetrical. It has no true tissues. It's referred to as a Perizone because it has no true tissues. And we should understand that it has no coelom, the locomotion, reproductive strategies, feeding strategy, and the various systems. Now, in order to understand how the Porifera feed and their gas exchange system, it's important to understand how the water is drawn into the Porifera. So, on our Porifera over here, we have these pores. On the outside of this sponge. Since it is porous and there are cavities, water is able to enter through those pores into the center over here. This central region is referred to as the spongocoel. The spongocoel is simply the center cavity. If we break down this word, we can understand that sponge, it's referring to our organism, which is a sponge, and coel means cavity. So we have the cavity of the sponge. Okay? So water enters into this cavity and it exits through the osculum. The osculum is simply a large opening in where the water exits. So water comes in through the pores, enters the spongocoel, and exits through the osculum. So how do we get this water rapidly coming in? This is when our choanocytes or collar cells come into play. So this is our choanocyte or collar cell, and we can see these finger-like projections, and we can see this flagellum. So these finger-like projections, essentially, they circulate the water and pull the water in and then back out. And when the water comes in, the choanocytes are able to engulf the food particles that are coming in via phagocytosis. Okay, so we have the water propelling inside because of the flagellating motion of these choanocytes. Water is being driven in, and then the food particles within the water are being trapped via phagocytosis by the choanocytes. Okay? Now here we can clearly also see the two layers. So we can see the inside cell layer is in contact with water and the outside layer is in contact with water. So all of the cells are able to diffuse and excrete through diffusion. Now if we go through each of these points, here we can see the Mesohill layer, which is right over here. The Mesohill layer, once again, is a gelatinous core that separates the two cell layers from one another. We have the epidermis, we have the pores from which water is entering, the spongocoel, the choanocytes, which will propel the water in and trap the food particles. And then we have the amoebocytes, which will take the food particles from the water and the choanocytes, digest it, and then transport the nutrients to other cells. So amoebocytes, they are a type of cell within the Porifera that are mobile, and they are mobile within the mesohill. So once again, within the mesohill, the amoebocytes are mobile and they transport the nutrients that they digest to other cells. Now, some terminology. Once again, hermaphrodites simply means that the organism is able to produce both sperm and egg, and it can self-fertilize or cross-fertilize. So if that organism is very isolated from other fellow organisms, it will self-fertilize so it is able to reproduce. But if there are neighboring organisms, what will happen is that the first organism will produce a sperm, and that sperm may swim to the other organism, and we'll see cross-fertilization. Another important thing to make note of is we just discussed that amoebocytes are mobile within the Meso Hill. How are they mobile? They're mobile because of their pseudopods. Pseudopods, essentially, if we break down the word, we know pseudo means false and pod means foot. So it's a false foot. And essentially what pseudopods are just the cytoplasm moving around to create these propagations, which not only allow the engulfing of food, but also allow that mobility. That is why amoebocytes are able to not only engulf food particles, but also move. It is because of these pseudopods, which is essentially just the distribution of cytoplasm poking out and helping that cell move forward. Now, the mesohill, why do we have that mesohill? It's not only an area for the amoebocytes to move, but it also acts as an endoskeleton and gives our sponge that tubular shape. And once again, it's that gelatinous region separating the two layers of the periphera. So we've talked about our amoebocytes. So they have that pseudopodia. It has many functions, including digestion and taking up food, not only from the water, but the choanocytes as well. They are able to move through the mesophyll, and they are totipotent. This is very important. Totipotent means that they can become other types of cells. So amoebocytes can give rise to other types of cells, and this gives our periphera a lot of flexibility, and that flexibility is very important because if you are in a marine environment, your environment is continually changing, and that flexibility allows the sponge to survive in various water conditions. Because the amoebocytes are totipotent, and if a certain cell is needed, the amoebocyte can give rise to that. The amoebocyte also makes spicules from calcium carbonate or silica, and these are used for support for the sponge. We discussed the pseudopodia already. It's extensions of cytoplasm used to pull the organism forward. And so we can see these cytoplasmic extensions coming out and engulfing this food particle over here. Budding we also spoke about, and we should understand what it means. It's when the parent organism produces a new organism via outgrowths. When the new organism is fully developed, it will break off. So here we have our hydra, and we can see this outgrowth occurring, and it's continuing to grow, and then finally, when it's grown enough, it will split off that parent organism, and now we have two hydras, and this hydra will continue to grow. And this is essentially everything we need to know about Phylum Periphera. The most important details are to understand that it is the basal taxon, it is asymmetrical, it is a Perizone, meaning it has no true tissues, and we should understand each of these terms: hermaphrodity, what mesohill is, and remember not to confuse this with mesophyll. The two important cells that we talked about are the amoebocytes and the choanocytes, which are both needed for our digestive process. And that is Phylum Periphera.